I'd like to return to the molecular world today and talk a little bit about DNA technology, some of the technology that's utilized with studies of the genome and so on. And so the first thing I want to talk about is a DNA fingerprint, because we hear a lot about that in the news, and there are a lot of tangible applications of DNA fingerprinting. So in DNA fingerprinting, really the first step is to take a DNA sample and cut it into segments that we call RFLPs. So again, as we've talked about DNA, I've mentioned several times that it's incredibly long. So when we look at one cell, the chromosomes in one cell would be about 3 billion base pairs long. So that's too long to manipulate, and we will actually have to be moving and manipulating some of the strands in this DNA fingerprint. So the first step then is to cut those incredibly long chromosomes into smaller pieces. And those smaller pieces are called restriction fragments, or RFLPs, but I'll get to that in a second. So the tool that we use for cutting the DNA is something called a restriction endonuclease or restriction enzymes. And they're actually a collection of these that we use. So what we do is we will take our samples of DNA, we put them into a small vial, add in one or more restriction enzymes, and then let them sit for a period of time, and the restriction enzyme will cut those up into fragments. So what I want to do is actually show you the nature of these restriction enzymes and how they work. And so I have a video that I can use to do that. So when we look at restriction enzymes and how they function, restriction enzymes basically will target specific sequences. So a particular restriction enzyme will target a very specific sequence of DNA. And typically what they're targeting are what we call palindromes, palindromic sequences. And these palindromic sequences, what a palindrome is that in English it would be a word that reads the same from left to right. So mom, for example, M-O-M in one direction, M-O-M in the other direction. That's a palindrome. And so again, when we're looking at restriction enzymes, they'll do this as well. So for example, if we look at this strand, we see the sequence, this would be 5' GAATTC. And if you look at the other strand, it would read the same in the opposite direction, GAATTC in the other direction. So a particular restriction enzyme, this one happens to be called EcoR1, named for the bacterium that it comes out of, E. coli restriction enzyme number 1, will target that sequence and then make a cut that's not a straight across cut, and we'll see in a second that it's kind of an angular cut. Now, a different restriction enzyme would target a different palindrome and also could make a cut that leaves what we call sticky ends. Some restriction enzymes will make a straight cut, but we're not going to really be concerned with those. I'm going to be interested in the ones that cut in this sort of manner. So again, let's go ahead and run this animation and see how this actually works. So again, the enzyme will target these palindromic sequence, attach, and then separate them. Now, what I also want you to pay attention to, we won't really be utilizing this today as we talk about a fingerprint, but it does come into effect when you talk about some other aspects of technology, is that restriction enzyme leaves what we call sticky ends. And so if you look at the ends of these DNA fragments, then you'll see that we end up with complementary ends. And this is going to be useful because these ends can be spliced back together. If we were trying to put unrelated DNA sequences together, we will utilize those sticky ends. So that's something that we'll actually be talking about in the future. Now, another example of a different restriction enzyme, again, it's going to make essentially a similar type of cut, but it's going to be targeting a different palindromic sequence. So if it'll come up here in a second. So again, now we're looking at a different sequence of bases, but a particular restriction enzyme would target that specific sequence, bind, and make the cut, again, leaving what we call sticky ends. And so these sticky ends, again, are complementary and again, won't really have any significance today, but will in the future. Okay, so once we have the cut then, what we want to do is actually separate those fragments of DNA. So again, the cut is actually going to produce different fragments of DNA. And ultimately what we're going to want to do is separate those fragments. And I'll come back to how we do that in just a couple of seconds. Now, there's something else I want to talk about related to restriction enzymes in producing these fragments. And it is the concept of what's known as a VNTR. When we look at the human genome, and if we're trying to establish genetic similarity between different individuals or between different samples. So in other words, we have two samples or more samples of DNA, and we're trying to establish if they're from the same person or the same animal. One of the things that we could target would be our genes, our protein coding genes. But there's not a lot of variability, for example, in the human genome. There's not a whole lot of variability when we target specific proteins. Typically, there's one allele or two alleles. Take something like hemoglobin. Almost everybody has exactly the same hemoglobin. So what we want to do then is target the so-called non-gene segments or the non-protein coding segments of DNA, because these tend to be more variable from person to person. So there's more variability there. That means more alleles, if you will, and it will be easier to tell if we have samples from one individual compared to a second individual, or establish relationships between individuals. So what VNTR stands for is it stands for Variable Number Tandem Repeat, which is a mouthful, but it explains really what we're looking at. So let's go ahead and look at a chromosome, and let's say that, or two chromosomes. Let's say that a person is homozygous for a particular VNTR. So what we're looking at is a homologous pair of chromosomes. Of course, chromosomes would be a lot longer than this. But what we're interested in here are the repeating sequences here. So again, we would be looking at parts of the genome that really are not coding for proteins. And in the non-coding parts of the DNA, what we tend to find are repeating sequences sometimes. So here we're looking at GC, GC, GC. Now, and this is the VNTR. So these repeating sequences tend to be variable in number, and they repeat. And we refer to them as being in tandem because your homologous pairs of chromosomes will each have those repeating sequences. Now, if you're homozygous for a VNTR, what it means is that you would have the same number of repeats on each of your homologous chromosomes. So in this case, we're looking at 3 repeats of GC. So in this example, a person has inherited 3 repeats from mom and 3 repeats from dad, and so this person is homozygous for this particular VNTR. Now, if we use a restriction enzyme then that targets this VNTR, so we'll cut on either side of the VNTR, what that will do then is create a fragment, or fragments of a particular length in this case. So if we target that with a specific restriction enzyme, again, in this case, targeting the GAATTC sequence, and we go ahead and let it make a cut then, what it will produce then is fragments, DNA fragments. And these fragments are called RFLPs, restriction fragment length polymorphisms. Of course, that's a mouthful, which is why we call them RFLPs. So they're also just referred to as restriction fragments. So again, by using restriction enzymes to cut the chromosomes, and by targeting VNTRs then, what we can do is produce fragments. And in this case, the individual is homozygous for the VNTR. So you'll notice that the 2 fragments are of the same length. So this will be significant later when we start to actually do the fingerprint. So if a person is homozygous for a particular VNTR, it will produce RFLPs that are exactly the same length. And again, that will then be significant when we run the fingerprint. So this individual is homozygous for the VNTRs, so fragments of the same length. Now, if we compare that to an individual, and again, so we have our fragments of equal length. Now, if we compare this to an individual that's heterozygous for the particular VNTR, again, if we target where the variable number repeats are, this individual inherited a chromosome from mom where there are 6 repeats, from dad 3 repeats, and if we target, again, that VNTR with restriction enzyme that cuts on either side, that will also produce fragments, RFLPs. But in this case, notice that if a person is heterozygous, it will produce fragments of different lengths. Now again, when we start to separate these out in a couple of minutes, that will be significant. So one individual is homozygous, producing fragments of equal length. One individual is heterozygous, which will produce RFLPs of different lengths. So that's an important concept to keep in mind. Okay, now, how do we separate the fragments into different lengths? So again, we basically have a tiny vial, and we've got these all mixed together, but we want to separate them out according to length, these RFLPs. So the process here involves gel electrophoresis. And so what we do is we'll transfer our DNA samples from small vials where we've mixed our restriction enzyme with our DNA sample. We'll load them into a gel composed of a material called agarose, or sometimes some other materials. And then we'll run those through a gel by using an electric current that will run from negative to positive. And then that will push the DNA fragments along. And in so doing, you may recall that diffusion of particles is not always the same. So particles that are lighter diffuse more rapidly than particles that are longer. So lighter particles diffuse more rapidly than those that are heavier. And in the case of DNA, then the shorter fragments will run faster than the longer fragments. And so they'll start to separate themselves out by length. So I want to go ahead and actually show you what these gels look like, although this is on a very small scale compared to what a working lab would do. So again, this is just kind of a classroom specimen that I'm going to be showing you here. So I first just want to show you the gel. And so the gel is kind of hard to work with, but it's not quite like Jell-O gelatin, but it's similar. And you wouldn't normally be touching this with your hands, but it makes it easier for me to manipulate. But you'll notice that the gel actually has indentations in it where we can load our DNA samples. And so we normally do our mixing of DNA samples in these small vials. And so then what we would be doing is using a pipetter, and we would transfer a DNA sample that uses this particular type of pipetter, and we would transfer those samples into the wells. Okay, so one sample would go into each well. And then we would load this, when actually, you don't actually load the DNA sample until it's in an electrophoresis chamber, but the gel actually goes in an electrophoresis chamber. And the electrophoresis chamber then also has to have the gel covered with some kind of electrolyte. It's a buffered electrolyte, so we'll cover that. And again, you wouldn't normally want to be touching this, but kind of need to work rapidly here. And so the real trick to this is that, again, your gel actually would be in a buffered electrolyte solution. And so what we actually have to do then is load our samples through the fluid and then fill the gels inside the fluid. So that's kind of the tricky part to this, because that does take a little bit of technique. So once we've loaded our DNA samples in, we would put a cover on this, we would attach it to a power source, and we would run an electric current through here, and that would basically push the DNA fragments from negative to positive poles. And again, as they run through the gel, they'll run at different speeds, and they'll slowly separate themselves out according to different sizes. So again, this is kind of a miniaturized example of what we'd be using in a working lab, but again, it works for classroom demonstrations. All right, so getting back again to the purpose of gel electrophoresis. The purpose of gel electrophoresis is to separate our restriction fragments according to size. And as they run, then they'll separate themselves out into bands, basically. So again, now also looking at this process diagrammatically then, so again, what we're looking at here is a gel. Here are the wells of the gel. This would be a side view of that gel, looking at the indentation in the gel. So again, we would take a DNA sample, load it into a well, and then we would load all of our wells in that way. And then again, this would go into an electrophoresis chamber. We would run the current, and the DNA would run at different speeds according to the lengths of the fragments. So again, a side view, they'll run out according to different lengths, and they'll separate themselves according to size as they do that. Now, even though this looks like a lot of fragments, the reality is that there would be thousands and thousands of fragments. It would be almost one continuous mass. But I'm representing them as separate lines just to emphasize the fact that each band then would have fragments of a specific length. Again, looking at a top view of the gel then, as we run the current through, the bands would separate themselves out. And normally what we do in the first well is that we'll put some kind of a control. We'll put a sample where we know the lengths of the fragments. And this will help us make sure that the gel is working properly and that things are moving at the appropriate speed. And then let's say here that we're actually doing a crime scene investigation. And let's say that we have 3 suspects, and then we have evidence from the crime scene here. And so again, we would load DNA samples from each of our 3 suspects. And then in our last well, we'll put evidence from the crime scene. And perhaps we're trying to link evidence from the crime scene to one of our suspects to place them at the crime scene. So again, the fragments then will run out of the gels, separate according to size. And again, there literally would be thousands of fragments. So one of the problems we have here is that if we were to stain the bands, like stain them to try to see where the DNA fragments are, there are so many fragments here that it would just be one continuous mass. So again, if you stained it, it would just end up being what we call a smear. It would just be a big block of stain or a big string of stain. We wouldn't be able to see where the individual fragments are because there are so many fragments. And so again, we now have to do something. We have to manipulate this in a way to try to figure out where the fragments of interest are to us. So the next step is to blot. And in the blotting technique, what we want to do is transfer the DNA samples from the gel, because as you saw, the gel is kind of brittle. It's not really very easy to work with. We want to transfer it to something that's a little easier to work with. And so again, after running our restriction enzymes and running it through the gel, we want to transfer this into something that's a little easier to work with, and that's known as a nitrocellulose membrane, or sometimes just called nitrocellulose paper. So what you do is you would have a container with an electrolyte fluid in it or an alkaline solution in it, and then a sponge on top of that, your gel on top of that, your nitrocellulose membrane on top of that, paper towels, and then something on the top so that it all gets blotted up from the gel into the nitrocellulose membrane. So it isn't always high tech, but the main thing is avoid contamination of DNA samples. So again, if we were to stain the gels, it would just be a big mess, a big smear. And so I'm going to kind of remove the smear to emphasize that we are dealing with fragments here. We would have thousands of fragments, but a lot of them. And again, what you would do is overlay your gel with this nitrocellulose membrane. So this would be from a top view. From a side view, we would overlay our gel with the nitrocellulose membrane. And then the fragments would diffuse from the gel into the nitrocellulose membrane, but they would maintain and do maintain their position, relative position to one another. So we transfer the DNA sample from the gel into something that's easier to work with, the nitrocellulose membrane. So again, looking at it from the top, then we would have overlaid our gel with the nitrocellulose membrane. And so where we had DNA samples in, those are now found in the nitrocellulose membrane. So this doesn't really solve our problem of where the fragments are, because the other thing you have to realize too is that the DNA is colorless. So really we wouldn't even be able to see any bands at this point. We wouldn't know where any of the fragments are. DNA is colorless. If we stained it, it would be such a dark and messy stain, we wouldn't be able to get much information from that. So we now need to try to figure out the genetic similarities and differences between our different samples. And how are we going to do that? The answer is we need to use what's called a probe. A probe is a short segment of DNA or RNA that's complementary to some gene of interest to us or some sequence of interest to us. So, for example, if you were looking for a particular sequence of DNA, you would generate a probe that's complementary to that. And the probe not only has to find it by reannealing with it, by being complementary to it and bonding with it, but it also needs to show us where it is. And so the probes that we use then will be either radioactively labeled, or they sometimes will have a fluorescent protein associated with them, so that if we want to find out where that particular probe is, it will tell us where it is. So again, we need not only something that will find where the genes are, but it will tell us, hey, here I am. The probe is right here. So again, just an overview of what we'd be doing then is the nitrocellulose membrane then is mixed with probes, and then the probes will find those particular sequences. And the way that they do that is, again, you'd put these in a solution that would cause the DNA strands to separate, and again, our fragments to separate. And again, you may recall that the VNTR we were working with was repeats of GC, GC, GC. So we would introduce a probe that would find that VNTR. And we would want to use short segments that would be shorter than the RFLPs themselves, than the fragments themselves, because we want the probes to diffuse more rapidly and associate with the complements before the larger strands can get back together, so that when they reanneal, they'll be blocked from reannealing. But the point is, if these are fluorescent or radioactively labeled probes, they can tell us where they are. So we can use those to show us where they are in the nitrocellulose membrane. So the probes will tell us where our VNTRs are. Now, if we go back to the idea of our RFLPs, targeting VNTRs. You may recall that these individuals were homozygous for a VNTR. And when we cut with restriction enzymes in, that produced RFLPs that were the same length. So when these run through the gel then, they'll run at the same speed. They will be found in the same band. And then when we probe for that particular VNTR, that would produce one band in the gel. If an individual is heterozygous for the VNTRs, and we probe for the VNTRs, and again, that would produce fragments that are of different lengths. So when we probe for the VNTRs, what we would end up with then is one fragment of one length, one fragment of different lengths. So this would be two bands in the gel. So again, the probe would find the GCs here and here. And so these would be running faster than these. So we would actually have two bands in that particular gel. So again, if we go back to our original gel, we've just got tons of bands, and actually these aren't even visible at this point. So what we would use then is a probe to try to find where our VNTRs are. So we introduce the VNTR. In this individual, we see two bands. So what that means is this individual is heterozygous for that particular VNTR, producing two different RFLPs running at different speeds. We only see one band here, so this individual is homozygous for that VNTR. This individual, only one band. Oh, excuse me, this individual has two bands, but they're at different places. So again, these would be VNTRs with differing numbers of repeats and fragments of different lengths. And here, heterozygous. So this individual is heterozygous, this is heterozygous. And if you look at this individual and this individual, you'll see the same banding pattern. And so that suggests that, hey, perhaps they're from the same individual. They at least share the same VNTRs for that particular gene. Now, could you say then that this sample and this sample are a match? Probably not at this point. So what we would want to be doing then is targeting other VNTRs. So we would cut with restriction enzymes on different sides of other VNTRs, perhaps a VNTR that's ATT, ATT, ATT. I shouldn't be advertising, I guess, but anyway, that's something you could target. So you would use a probe to target that VNTR where you've cut on either side and produced restriction fragment length polymorphisms, RFLPs, that are different fragment lengths based on those VNTRs. And so again, if we probe for that, again, we would have an individual here that's homozygous for those particular VNTRs, heterozygous for the VNTRs, heterozygous for the VNTRs, and homozygous for the VNTR. Now, again, we're seeing that this individual and this individual match up again. So now we're beginning to think, hey, maybe these two are from the same samples. Now, what I do want to say about this process is that DNA fingerprinting can never, with 100% certainty, tell us the two samples are from the same individual. We can be pretty confident, but we are never going to be 100% confident. But what we can do with 100% confidence is exclude individuals. So these two individuals we can already exclude from the crime scene from the crime scene DNA at least, because they don't have the same VNTRs. And so if they don't have the same VNTRs, they're not genetically identical to the DNA from the crime scene. If you're not genetically identical to the crime scene DNA, you can't be the same person from the crime scene. So ultimately, DNA fingerprints are more exclusionary than inclusionary. So again, this increases our certainty that these are from the same individual, but let's go ahead and introduce still another VNTR and a probe for a particular VNTR. And again, we can see individual heterozygous, homozygous, homozygous, and heterozygous, and that's increasing our certainty. And still another would increase our certainty even more as we see matches. So once we've probed with our VNTRs then, really ultimately it comes down to reading our gels, or reading, in this case, reading our nitrocellulose membrane. So we read the fingerprint finally. So again, it's the probes that's actually telling us where the VNTR is. Up to this point, we don't even know where they are because they're colorless and we haven't stained for them. So we would ultimately then be left with a banding pattern. And if we go ahead and get rid of all of those except those that match, we do see that we have a match with our crime scene to one of the suspects. So now the question is, well, how certain can we be that they're the same individual? Well, again, we can never be 100% certain. But what we would do then is begin to look at this. Hey, it's amazing that they were the same for one gene. What would be the likelihood that they're the same for 2 genes and 3 genes and 4 genes? And as we've gotten more experience, we've been able to predict this a little bit more. So let's say that in our example here, that the first VNTR, if you shared that gene pattern with another person, that would happen in 1 in 5,000 times. So there's only 1 in 5,000 chance that you would be identical to another individual. And let's say that our second VNTR, the chance of being identical to them would be 1 in 20,000. And our third VNTR would occur only 1 in 3,000 times. And our fourth VNTR, 1 in 30,000 times. Any one of those, you might be somewhat skeptical that perhaps they're not from the same individual. But the fact that they're going to follow the random laws of probability, what we can do is multiply these independent events to get the combined probability. So the probability that 2 individuals would be identical for all of these VNTRs would be an extraordinary number. What, 1 in, 9 quadrillion? That's almost like an imaginary number. So again, we can't say with 100% certainty in this case, but boy, we can be pretty certain that these 2 samples are from the same individual. So the standard that the FBI uses for considering 2 samples to be a match and from the same individual is 1 in 260 billion. And given that there are only about 7 billion people on Earth, that's a pretty high standard. So again, from an investigatory perspective then, you can never be 100% certain, but the more VNTRs you probe for, the more matches you have, the greater the certainty that samples will be from the same individual. But what you can be certain of is when you don't have matches, you can be 100% certain that they are not from the same individual. So when we look at DNA fingerprints, there are just a multitude of uses for DNA fingerprints. So certainly crime scene investigations, as we've talked about, establishing paternity. Anytime you want to establish the genetic similarity or differences in samples, it comes into play. Ecologically, we've been able to use DNA fingerprints to tell us a lot about behavior in animals. So for example, those animals that form harems, that have lots of females, we can now actually, in the past we would have had to look at mating and see who mated with who, but now we can actually look at the genes, because what we have found is that sometimes the so-called harem master, who's controlling a harem, a lot of times he isn't 100% successful. Other males get in sometimes and actually mate with the females. And then also it's a really good way to do population studies. By just taking fur samples, for example, or feather samples, you can do DNA fingerprints, you can trace where individuals are moving within a particular ecosystem without ever seeing the animal itself or ever having to handle the animal. Okay, well, I'm out of time, so that'll do it for today.